Exercise 13 deals with neuron anatomy and physiology. With the nervous system, when we are looking at the different types of cells that are present, you have the neurons, which is what most people think of, but you also have a lot of supporting cells, and these are known as your neuroglial or just glial cells. And within the nervous system, you've got the central nervous system, and then the peripheral nervous system. There are different types of neuroglial cells depending on which of these systems you're talking about. In the central nervous system, there's 4 different types of neuroglial cells. You have the astrocytes, oligodendrocytes, microglial cells, and the epidermal cells. But in the peripheral nervous system, there's only 2 types, the satellite cells and the Schwann cells. In this diagram, it is showing some pictures or schematic drawings of some of these different types of cells. Up here you have the astrocyte in the central nervous system. Here is your neuron in yellow. And around the capillaries, you have the astrocytes, which are attached. Basically, they are involved with the blood-brain barrier in terms of providing extra protection to the neurons in the central nervous system, in the brain, the spinal cord. Essentially, anything coming out of the capillaries have to go through the astrocytes to get to the neuron. It's just an extra layer of protection. Your microglial cells, as you can see here, look very kind of spiny, helping to form the scaffolding between neurons. You have your epidermal cells, and then the oligodendrocytes. They wrap around the neurons, providing insulation, helping to provide this myelin sheath that we see. Excuse me. And then with the peripheral nervous system, you have the Schwann cells and the satellite cells. The Schwann cells are similar to the oligodendrocytes from the standpoint that they also will form this myelin sheath and wrap around the neuron. With the oligodendrocytes in the central nervous system, it's one cell that wraps around the entire neuron, versus in the peripheral nervous system, each one of these small little bundles is an individual Schwann cell. So peripheral nervous system, once again, satellite cells helping to support the cell bodies, and then the Schwann cells are helping to form those myelin sheaths. Similarities, but also differences to the oligodendrocytes, as I just said, from the central nervous system. Those microglial cells, they're going to be involved with also protection, involved somewhat with the immune system, helping to remove any type of debris that is present. Astrocytes have numerous jobs, once again, helping with that blood-brain barrier, helping to secrete different factors, helping to take up various things such as potassium and water and neurotransmitters. And then your epidermal cells, this is also helping to perform or create barriers between different compartments. So they're all helping to assist the overall functioning and help improve the functioning and improve the efficiency of the neurons. So the neuron itself then is the basic functional unit of the nervous system. It's going to help to transmit these electrical impulses, which we call the nerve impulses. Oftentimes we'll talk about gray matter versus white matter, etc. Gray matter is usually the cell body, the main portion of the cell. This does not have either the oligodendrocytes or the Schwann cells wrapped around it. And so we'll see later when those supporting cells are wrapped around the appendages or extensions of a neuron, tends to give it a whitish color. But the cell body tends to have more of a grayish color, so we refer to it as gray matter. Now, what do we do? Yes, we have different terminology for you, and the cell body, when it is in the central nervous system, when you have a cluster of those cell bodies or a group of them in the central nervous system, so the brain and spinal cord, we will refer to that as nuclei. We're not talking about the nuclei of cells. It's just that you have a group or a cluster of these cell bodies. When we have a cluster or a group of the cell bodies, but it's in the peripheral nervous system, meaning outside the brain, outside the spinal cord, we're going to give it a different name. And that name is going to be the ganglia. This way you can distinguish when discussing with someone, distinguish whether you're talking about central nervous system or peripheral nervous system when you are talking about these groups of the cell bodies. So nuclei in central nervous system, ganglia in the peripheral nervous system. I'm sure you're thinking that it is some ulterior motive to annoy anatomy and physiology students by giving you this vocabulary, but it does help you to distinguish between them. Dendrites are the receptive regions, so they are receiving the impulses, and the axon are those extensions that are, the impulse is generated and then it's moving away from the neuron typically. It's moving away from the cell body. The axons typically are longer than dendrites. With the neuron, there's different regions. The axon hillox, this is an enlarged area of the cell body. It's at the proximal end of that axon. So just as the axon, basically think of where it's attaching to the cell body. The axon terminus is at the very end of the axon, the very distal end of it. It does contain a lot of vesicles and inside these vesicles are the neurotransmitters. And then the synaptic cleft. This is a tiny space between your presynaptic neuron and the postsynaptic neuron or the muscle. So these smaller extensions up here are the dendrites. As I said, these are the receiving extensions, the receiving impulse from another neuron. Here is the cell body, which we call the soma. So yes, again, we're going to get different names. Just this would be the nucleus right here. This enlarged area kind of, excuse me, kind of looks like a funnel, but it's where this long extension is the axon. So it's where the axon attaches to the cell body. So it's that widening, I often say it looks like a funnel. That's the axon hillox. So you generate the impulse or receive it here. It's going to now move down away from the cell body, move down that axon. These lighter colored areas, that's the myelin sheath. So if you were talking about the peripheral nervous system, these would be Schwann cells. They help to insulate because this electrical impulse, it's an electrical charge. It's just like, think of electricity passing through wiring at your home. You want it to be insulated so it doesn't spread all out in the surrounding areas. It keeps it contained, moving down that axon. So that myelin sheath helps to insulate. And then this area down here is the axon terminus, the very end. And if you enlarge those, you'll notice that, yes, it is containing several vesicles, these little sacs, and inside those are the neurotransmitter. And then that is going to be released when the electrical charge comes down. It's going to be released, and then it's going to stimulate whatever this is connected to, either another neuron or a muscle. So the synaptic cleft is this tiny space. It looks large here, but it's actually very, very small. The presynaptic, the one that the charge is coming down, and the postsynaptic, the one that's going to receive the charge, are not physically touching each other. So initially this would have been the presynaptic. The charge came down, it was transmitted and stimulated the dendrites. So initially this was postsynaptic. Now it becomes the presynaptic and it's going to stimulate over here. That myelin sheath, as I said, those axons are going to need insulation, and that's what the myelin sheath is. So most of your axons have a myelin sheath around. There are a few that do not, but the impulse, that electrical charge, will move much faster if there is a myelin sheath. So if it has that insulation covering, it does move much, much faster. Once again, oligodendrocytes in the central nervous system produce these myelin sheaths, as you can see here, and then the Schwann cells produce the myelin sheaths in the peripheral nervous system. Now, neurons, you can classify them one of two ways, either by structure or by function. If you classify them by structure, excuse me, This is showing several different names. Anoxic neuron. You have several different extensions or processes, but you really cannot distinguish the dendrites from the axon. They all look the same size. A bipolar neuron, you have the dendrite, the cell body or the soma is in the middle, and directly opposite is the axon. So you have just two processes and the cell bodies in the middle. Now, a unipolar or pseudo-unipolar axon, here the cell body, it may look very similar to a bipolar, but the cell body is actually off on the side. And the dendrite is really continuous with the axon versus being separated by the cell body. And then a multipolar neuron, these have several different processes. They tend to have a single axon. Now you may have branching at the terminus end, but you have multiple dendrites. The majority of the neurons found in the human body are going to be multipolar. Bipolar ones are typically seen in the eye with part of the retina, which is helping you to see, basically absorb the impulse and then send that impulse via the optic nerve. If you classify neurons according to function, you classify them obviously by what they are doing. And so you can have sensory neurons, which are also known as afferent neurons. They are receiving an impulse from a sensory receptor, and then they're sending it towards the brain or towards the spinal cord. They're sending it to that central nervous system. Interneurons, also known as association neurons, they're the intermediate. They're the middleman. They will connect a sensory neuron to the motor neuron. And then the third type is the motor neuron, or the efferent neuron. They are receiving that impulse from the inner neuron and sending that impulse then away from the central nervous system. So a sensory neuron sends impulse towards the brain, towards the spinal cord, the central nervous system. Here's the incoming information. The interneurons or association neurons have to decide, okay, what do we do with this? Do we do anything with it? Sometimes the impulse just stops there. If you need a response, then let's say you put your hand on a hot burner. That sensory receptor picks it up, activates the sensory neuron, sends it to the central nervous system with that information going, ooh, my hand is hot. The interneuron or association neuron interprets that information. Do we do anything? Well, yeah, we kind of need to move the hand. So it's going to send that information down the motor neuron away from the central nervous system, and it's going to stimulate the muscle for you to contract the muscle and move your hand away from the hot source. And that's what this diagram, excuse me, is showing. You have the sensory neuron. Here it becomes stimulated, it moves it. Now, this is just showing one nerve, but you're actually going to have a whole series of nerves coming up. Ganglion, remember, that is going to be where you have a group of cell bodies, so not just one, there's going to be a whole group of them here. That sensory neuron comes in. So all of this is the peripheral nervous system until you reach the spinal cord. Now you're in the central nervous system. Here's your association or interneuron. Process information. It may send information all the way up to the brain. It may just stop in the spinal cord. It depends on that individual circumstance. The interneuron stimulates or activates now the motor or efferent neuron, which then sends the impulse away from the central nervous system, and down here it's going to be stimulating either another nerve, a muscle, or gland. So with the central nervous system, when you have a whole bundle of axons together, we call that a tract, a nerve tract. In the peripheral nervous system, when you have a whole bundle of axons, we call that nerve, like spinal nerve, cranial nerves. Sensory nerve, once again, is going to be sending impulses to the central nervous system. Motor nerve sending impulse away from the central nervous system. Now, sometimes you may have a mixed nerve, and that just means that it contains both sensory and motor fibers in there. So both sensory and motor, those fibers, those axons, are both in there. If we look at the structure of the nerve, you have different layers, if you will, of connective tissue. And we saw part of this when we looked at the bone with the periosteum and endosteum. And in some ways, it's very similar. You also saw it with the little bit with the coverings of the muscles. Now, again, with the nerves, if we move from the interior and move more superficially or outward, the endoneurium is a connective tissue that's surrounding each axon. So it's surrounding outside the myelin sheath. Perineurium is when you have connective tissue that's surrounding now a group of axons that's called a fascicle. And then the epineurium is when you have connective tissue that's surrounding a group of fascicles. So you're getting larger and larger and larger. So on this diagram, if we look here, this yellow down here is the nerve fiber itself. So now you have the whitish color is the myelin sheath. If this were a peripheral nerve, it would be the Schwann cell that's wrapped around, providing that insulation. Around that, on the outside of that, so think of you've got a shirt on, you're chilly, so now you put a sweatshirt on top. That would be the endoneurium. That's the slider color. You package this together. That group or that bundle is now wrapped by connective tissue called the perineurium. And this is called a fascicle, this group of them. So now it's like, here you are, you were cold, you, outside of your shirt, you put a sweatshirt on. You're at, prior to COVID-19, you're at a ball game, you're with your friends, you've got your little kids or nieces and nephews or whatever, and everybody's cold, so you have a huge stadium blanket and you're wrapped with that. That would be the perineurium. You get a bunch of these fascicles grouped together, and then that is wrapped with the connective tissue called the epineurium.